Can anybody please confirm that you can see our screen?
Looks good.
All right. Sweet. Okay, I'm gonna try to split my attention between the screen and the people in the room. So apologies if I'm not always looking at all of you. It's nice to be here today. I love talking about the work that I do at King County. So thank you for listening to me talk about the work that I do at King County. I saw some colleagues on the line already. Special welcome to all of you, who I'm sure some of this will not be new for you, but today I'm going to talk about long-term monitoring in Central Puget Sound and get into some stories of past and present water quality. Okay, let's see. Next.
You will have to come.
This way you get to see my cursor as I dance across the screen. I'll try to keep that distraction to a minimum. So here is the plan for my talk today. So I'm going to walk through the monitoring that we do in King County, in Puget Sound, talk about some multi-year trends. This will be a lot of proselytizing about and selling our data set in a sense, which will be hopefully useful to some of you at some point who did not know that this data existed. I'm going to get into some cool stories from the data, so it's the past part, and then also talk about some present issues in Puget Sound, namely nutrients, quantifying wastewater impacts, and ongoing research, which is most of my day-to-day, but also has some fun tie-ins to my former academic life when I thought a lot about nitrogen, but sort of in a different context. I never envisioned that it would be so deeply involved in wastewater, but here we are. All right, so our monitoring program in Puget Sound comprises 4 main groups. So we've got water quality monitoring, both offshore, so in deeper water, and also closer to shore at beach sites. So the water quality sampling is most of what I'm going to talk about today, because that is mostly what I work on. We also have some food web monitoring, in which we collect phytoplankton and zooplankton samples, and then also some sediment monitoring, again at deep and beach sites. There are staff from the Science and Technical Support section, which is what group I'm in at King County, that manage all of these programs. Most of the samples are collected by staff at the King County Environmental Lab, which is located sort of on the south side of the Montlake Cut, just across the water from Fremont. We've got a research vessel that I'm going to talk about in a little bit, and field staff that collect all the samples, and then staff at the lab who process most of the samples, with the exception of the zooplankton. So this is the who and the what, and now we're going to get into the where. So we're going to talk about where we are sampling in Puget Sound. So we're focused mainly on the central basin of Puget Sound, which is where King County is located. So you've got a whole bunch of different stations that are shown here on this map. The purple dots are where we collect water quality samples. The little green cylinders are where we have some moorings that are collecting continuous data. I guess they're like gray. I didn't make this map, so I didn't choose these icons. These gray stars where we collect phytoplankton, and then the little copepods, the green copepods, are where we collect plankton samples. The sampling frequency is roughly twice monthly during the growing season, so from February to November, and then monthly from December to January, when much less is happening in Puget Sound. You'll also note that there are 4 stations in the Duwamish River, and you might be wondering, why are these included within a marine monitoring program? This has been a point of contention for a while of who owns these stations, whether it's our freshwater monitoring group or our marine monitoring. Right now, we own them in the marine group, even though they're fresher than the rest of the data that
But
we
I guess
collect.
the 2 down beach, very close to Harbor Island, that's still marine for.
Yeah, it's very salty. Yeah, you still get a prominent salt wedge at all 4 of these sites, though perhaps the least, obviously, at the one that's furthest up the Duwamish River. These stations were selected, most of them a couple of decades ago. Some of them, the data goes back all the way to the 70s, and some even before that. But there is a mix of what we consider to be ambient stations, and then some that are closer to outfalls, whether that's outfalls of wastewater treatment plants or wet weather treatment plants, which are now circled in the red and yellow. So there's a good mix of stations between things that are closer to wastewater treatment plant outfalls, but also some that would be considered more ambient. The station up top here is close to our Brightwater outfall. But the station I'm going to talk about the most today is the one just south of that. This is our longest term station, KSBP01. The naming conventions are based on a map that no longer exists, so people can't find. So all of our stations have really horrible names, but that is the one that you are going to see the most today. Point Jefferson, aka KSBP01. Okay, so we've gotten through the who, what, and where, and also when, but now the why. Why are we monitoring Puget Sound? And specifically, why is King County monitoring Puget Sound? It's pretty unusual for a county level jurisdiction to have this much monitoring infrastructure. We have we have 5 scientists on the marine team at King County working on these monitoring programs. So it's something like 45 scientists in the science and technical support section who do a lot of different types of monitoring. So it's pretty unusual. And I guess the question is why? So the things that we've been tasked with in our group, one thing is to characterize water quality in food web conditions. This could mean a lot of things, but primarily what it means is that we report regularly on the things that we are observing in Puget Sound. We're not necessarily trying to ascribe causes, though sometimes we do, but more often than not, we are simply reporting on what we are seeing and the conditions that we are observing. We are also evaluating trends. One of the benefits of having a very stably funded monitoring program is that we are able to measure the same things over a long period of time. So we can really say with confidence whether things are changing or not. Some of our programs, namely the sediment monitoring program, is also contributing to National Pollution Discharge Elimination System, or NPDES, requirements associated with those wastewater treatment plants. The water quality monitoring that we do is entirely separate from this. It's not actually tied to any permit requirements at all. It's just the sediment monitoring that's tied up in that. And then finally, one of the things that we do as part of our programs is to assess impact of events on water quality. So for example, when West Point flooded, we were able to say with the data that we have been collecting and data that we collected after the event, whether we saw a change in water quality as a result of that event. So these are the primary functions of our program. And now we're going to move on to the how of our program, which is primarily through our research vessel, the Sound Guardian. I think the name was chosen because it sounded like Sound Garden. I think Sound Garden was actually there when the boat was christened in 2016. It's a 48-foot aluminum catamaran that is housed there at the environmental lab on the Montlake Cut. And this is the vessel from which almost all of our samples are collected, except the ones that are in very shallow water and have to be sampled from shore. There's a big A-frame on the back of the boat here. This is what we use to do the bulk of our monitoring. Underneath the A-frame is the CTD rosette, which I am going to talk about next. I know that some of you probably have a little bit of familiarity of how oceanographic samples are collected, but for those of you who don't, you're in for a real treat. You're going to learn a lot about it in a really short period of time. The primary way in which we collect samples is through a CTD profiler and Niskin rosette. So here I'm going to be talking about the CTD profiler, which is circled in orange here. This is a system that is attached to the Niskin rosette. And as we lower it through the water, it is collecting continuous data of a variety of kinds. It is collecting temperature, is collecting salinity, is collecting pressure or depth, is also collecting dissolved oxygen, nitrate and nitrite concentrations, chlorophyll, fluorescence, and light. So you get a profile all the way down and all the way back up with this instrument. And the data looks something like this. So here is where KSBP01 rears its head. Here is data from October over a variety of years. Each profile is shown. You can see it changing over the depth. This is one of the deepest stations in Puget Sound. It's really quite deep. Puget Sound is a fjord-like estuary, and so it's very deep, and this station really characterizes that well. What we're looking at here is dissolved oxygen, so that's what's on the x-axis, and then depth obviously on the y-axis. The top of the water is here, going all the way down to the bottom, just about 270 meters. One of the things that I love most about this data is that it really highlights how changeable things can be in the middle of the water column. We can look out at the sound and we can speculate about what's happening underneath it, but until we're actually putting instruments out in the water and dropping them further down from the surface, we really have no idea what's happening at depth. I guess another point I wanted to make about this data is that these are profiles from pretty much all October profiles that have been collected at this station. The Washington water quality numeric standard for the Central Basin of Puget Sound is 7 milligrams per liter dissolved oxygen. You'll note that every single one of these profiles is below that numeric water quality standard. We will come back to that later. Here's another way that we like to visualize this CTD data. This is a contour plot, again, KSVP01, from 2022, showing how dissolved oxygen changes both over time on the x-axis and depths on the y-axis. This is just the top 30 meters of the water column, but I think it still illustrates the point that things change in time and they change at depth. This black contour line is that 7 milligrams per liter, which is that water quality standard. And you can see that even at the surface, it's often below that water quality standard. You'll see some sort of light purple spots at the top here, especially in the spring and summer when you have a lot of phytoplankton growing at the surface. They cause the oxygen to be quite high when they're growing. Obviously, they're producing oxygen during the day. And then consuming it at night, but we only sample during the day, so we get very high dissolved oxygen at the surface. Each of these black lines vertically is one of the sampling events. So this is just a filled contour interpolating, you might say, the changes over time.
I have a question.
Yeah.
So that contour line is kind of showing that all the values inside that contour line are below 7.
Right.
So from August to December, we are seeing a lot of below 7.
Yeah, what happens in the
Can you repeat the question?
Sure. Yeah, the question was whether every single observation within this black contour line is below 7 milligrams per liter. And the answer is yes, that's what that is showing. So what happens in the fall and early winter in Puget Sound is we get a lot of very low dissolved oxygen water coming in from the Pacific Ocean. And much of that water enters sort of deeper in the water column, but it finds its way to the surface during those months. So even in this top 30 meters, you can see that the dissolved oxygen concentrations are getting quite low, that there's not so much replenishment of dissolved oxygen at the surface, and it can't make up for those very low values in the fall. Okay. So in addition to the CTD data, we also collect bottle data. So these bottles are called Niskin bottles, and it's on a Niskin rosette. So what happens with these bottles is that as this entire carousel is lowered through the water column, we're watching the data come in through the computer that's on the boat. And when we get a sense of what the water column looks like as we send the carousel down, and on the way back up, we can snap those Niskin bottles shut. So there are little cables attached to each of them. So we can snap them shut at particular depths. So we usually have a number of set depths that we are targeting, say like 5 meters, 25 meters, 55 meters, etc. It depends on the station. But one of the things that we also try to target, the chlorophyll max. So that's the point in the water column at which chlorophyll is the highest. There's hypothetically the most biological activity at that point. And we try to collect an extra sample from an extra Niskin bottle at that chlorophyll max. So one of the benefits to having this bottle data, in addition to those beautiful CTD profiles, is that they're oftentimes more accurate than the values that we can get on the CTD. We can measure almost as many things as we want. It just depends on the volume of water collected in each of these Niskin bottles, but we tend to measure nutrients, so nitrate, nitrite, phosphate, silicate. And we can also get a more quantitative value for chlorophyll A and also its sort of byproduct phaeophytin A. But these values, specifically nitrate and chlorophyll, are much more accurate than the sensors, which I'm going to show on the next slide. So this is a profile, again, from beautiful KSB 201, again in October, looking at nitrate and nitrite. So all of the sensors, in addition to the way that we measure it in the lab, measures nitrate and nitrite combined. Nitrite is very low in Puget Sound. So really, it's just nitrate, but for the sake of it, I'm going to include them both because nitrite is very near and dear to my heart. I think it ought to be included. So in the black, we've got the CTD profile of nitrate and nitrite. This is using a STUNA instrument from Seabird Scientific based in Bellevue. You can see that it's very messy. This is characteristic of the STUNA data. One of the things that we like to do with this data, after we also collect bottle samples, which are shown in the orange here, is we're able to correct this whole profile of data based on the bottle samples. There's usually some sort of bias in the CTD profile, and it's very nice to have this bottle data as sort of a ground truthing, but also it helps us create a better data product up the other side. All right, the third and sort of final data stream that I'm going to talk about are our moorings. Shown now, I've updated the map. They are the sort of squished smiley faces. So we have 4 moorings in Central Puget Sound. These are collecting data every 15 minutes. It reports in near real time. The data gets collected by the instruments and it gets sent via cell signal to a third-party service, and then it gets sent to a King County website from the third-party service. It's very convoluted, but the point is that if you wanted to look up what water temperature is like at the aquarium, just near the Fauntleroy Ferry Terminal or in Quartermaster Harbor on Vashon Island, you can get that information right now on the King County website. I should also make a plug and say that all of our data is publicly available, which is really, really nice. I love being able to work at a public institution where I can just share data with anyone who asks for it. So if you want to ask me for data, you can ask me for data. But this data you can usually just get yourself if you really wanted to. At most of these moorings, so the samples are usually collected every 15 minutes, usually at one depth, except for the Seattle Aquarium. So this is a system that's sort of stuck on the side of pilings at the aquarium. It's collecting data at 2 depths, at the surface at about 1 meter, and then again at 10 meters. It's really interesting, especially because it's so close to the mouth of the Duwamish River. You really see quite a difference between that 1 meter and 10 meter, especially in salinity. Much pressure at the surface than it is deeper, which is kind of interesting. One of the many joys of having these instruments deployed continuously is that you get a substantial amount of biofouling. The Puget Sound is a very productive place. So on the left, you've got a new instrument that hasn't been stuck in the water yet. And on the right, you have an instrument that has been out for 3 months. This is what it looks like after 3 months of deployment. It is insane. You get such weird things happening in the data. Sometimes small jellyfish or other things will be sucked into the intake, and it really just wild things to the data. There's this little solitary tunicate. It's like a little globular thing that really likes the Teflon junctions in our pH sensors. And so you can see the pH will take a nosedive. It's like, my little friend is back. And we have to go, someone has to go and pick this little glob off of the sensor in order for the data to be restored. So there's a lot of problems with this data, but having higher time resolution data can be really useful to understand dynamic ecosystems, like in Quartermaster Harbor. Quartermaster Harbor is sort of a shallow embayment between Vashon and Maury Islands. And this data is from Quartermaster Harbor, the yacht club, which is all the way at the end of the harbor. It's probably about 6 or 7 meters deep at this spot. It's really mucky at the bottom and it gets quite anoxic. And so here we're looking at about a week of dissolved oxygen data. I think this is from 2022 again. And you can see that dissolved oxygen changes quite a lot, even over the course of a single day. This is the spot in Central Puget Sound where oxygen tends to get the lowest. You can see that it is approaching zero at quite a few points in time. This is very, very regular. It does this every fall. Look at this difference. That is, going from near zero hypoxic to super saturated in dissolved oxygen within a single day. There have been points in time at this location where the oxygen has been so high that there have been bubbles at the surface. And I dearly wish that I had a video to share with you, but the field tech who saw the bubbles did not take a video, for which we will forever resent him. One of the other joys of having these moorings deployed is that it can capture discrete events that we don't plan for. In this case, the tsunami that happened off the coast of Japan in 2011. This is from the Docton Park mooring that's in sort of outer Quartermaster Harbor. This is attached to the bottom of a piling on a dock. So you'll see that the depth is changing with the tide, just the amount of water that's over the instrument. And things look very smooth, and suddenly they look a little wiggly. This is an effect of extra water sloshing into Puget Sound from the tsunami. I think that is incredible. We would never have been able to see this if we didn't have these moorings already deployed. One of the other things that I wish I had interesting data to show you from this event was when the pier collapsed next to the Seattle Aquarium, probably a couple of years ago. They were doing some sort of construction to remove it anyway or renovate it in some way, and it just all collapsed into the water. And because we had instruments right there, we were able to see almost no change in water quality from that pier collapse, which was unexpected, but also reassuring because I would have at least expected some change in turbidity at least, but we saw almost nothing, which was good to know, I guess. Okay, so that is the long and the short of, mostly short, I guess, at this point in time, of all of the different types of data that we collect. Any questions before I get into sort of the more stories over longer periods of time?
Is 3 months the lifespan of that sampling? Is that more in sampling need?
No. So each, that would be awful. They're so expensive. Yeah.
Did everybody get the question? Do you want me to repeat it? The question was if 3 months is the average life of that
Instrument.
instrument. Yes.
The answer is it's not the lifespan. It's how long it should be deployed before the data starts going really, really sideways. They'll need to be taken back into lab and calibrated and cleaned, and then redeployed. We try to swap the instruments out after 3 months, just because that is a good period of time where the data really doesn't suffer data quality anyway. But sometimes, like in 2020, when we weren't able to do as much work in person, the instruments were out there longer, and it was fine. The data was fine. But thankfully, the instruments do get redeployed after some gentle calibration. All right. Well, with that, I'm going to get into some multi-year trends and other assorted stories. Just kind of a random, maybe not random, a smattering of different stories using different data streams before I get into the nutrients, which is really the meat of what I do day to day. Okay, so first I'm going to show you something that was published recently, not in a paper, but in what we affectionately refer to as the Big Ass Report. Probably shouldn't be saying that.
Yay report.
It's a large report, reporting on 2008 to 2019. So this is the span of time in which no reports have been published about any of our marine data. So we were playing catch up. This is data from, again, KSBP01 at the surface, looking at temperature anomaly. So red bars above zero, positive anomaly, blue bars below zero, negative anomaly. Superimposed are the ENSO and PDO, so the El Nino Southern Oscillation and the Pacific Decadal Oscillation climate indices in the solid and dashed lines. One of the things that was not, I guess, a surprise, but reassuring to see in this report is that there is quite consistent tracking between things that we observe in Puget Sound, like temperature in particular, and these regional climate indices. Very cool stuff. You'll note this sort of extended period of red bars. This was a marine heat wave. So starting in about late 2013, early 2014, out in the Pacific Ocean, there was the blob. There was the persistent warm anomaly in the water. And we really didn't start seeing a signal from that in Puget Sound until end of 2014, all the way through 2016. So because the residence time of water in Puget Sound is quite long, we saw sort of lingering effects even after the blob had dissipated in the Pacific, that we still saw elevated temperatures here in Puget Sound. And it was really, really hot in the vast majority of our stations, with the exception, oddly enough, of Quartermaster Harbor, which this sort of shallow embayment, you might expect it to be even hotter, but because it's already so hot, nothing really changed. It looked pretty normal, just kind of not expected. You might ask then, how is temperature, we can see that temperature changes in response to these climate indices, but how is temperature changing over time? So this is the full reliable data record, again from Point Jefferson, aka KSPP01, at now at depth. So this is deeper water, maybe not as changeable day to day due to solar radiation or whatnot. So this is really capturing how hot is that water over time. And you might say, well, it kind of just looks like a mess of data points, Taylor. I don't really see much of a trend in there at all, though you do see the really high, low values in those marine heat wave years here. One of the things that we did as part of that large report was that we calculated some trend analyses using seasonal Mann-Kendall analyses. And we did that over a couple of different time spans. The first of which, oh, there's my marine heat wave. Yes, marine heat wave, there it is. The first analysis we did was over that report timeframe, which was 2008 to 2019. And we found a trend of a warming trend, specifically a very significant warming trend, all stations, all depths, of about 1 degree centigrade per decade, which is really alarming. You might say that's really hot. That's far outpacing sort of the global temperature change of about 0.2 centigrade per decade. And I will say, yes, that is alarming, in part because there is the marine heat wave here at the end of that data record. That seems about right. So when we expand the trend analysis to go a little further back, we see that it lines up really nicely with sort of the global temperature change that we're seeing in the global ocean. Very similar to what we're seeing in Puget Sound.
I do have a question.
Yeah.
Those are trends analysis only performed over 2 decades because the other data are not reliable, or because we don't have prior data?
That's a great question. I would say that the older data is not as reliable. So
We have to wait for more decades.
that's right. Yeah, so we just have to keep collecting the same old data in order to really get a updated and continuous trend analysis. This is me plugging
PDSP01.
yes, KSPP01, our longest time record. Also, please pay your taxes because they paid for this program. Okay, another fun story. And this one is about, this is another plug for monitoring things over time. Our monitoring capacity has changed quite a lot over time. We are better at seeing things now and measuring things now than we were decades ago. What we're looking at here is Noctiluca. This is a dinoflagellate that's shown at the top here, a lovely microscope image from my colleague Gabriella at the Environmental Lab. And this lovely little dinoflagellate causes what we might lovingly refer to as tomato soup blooms. So that's what you're seeing in those 2 pictures, is the lovely, just red, thick algae blooms. These have been happening, as far as we know, all the time in Puget Sound since before our monitoring program started. So that's what you're looking at. On the left-hand side is a photograph, an aerial photograph from June 1975 with the Noctiluca plume. And on the right, another aerial photograph, June 2018, also a Noctiluca plume. We are just much better at seeing them, measuring them, finding them now than we used to be, which can be a little freaky because people are like, oh man, is this happening more? And the answer is we don't really know because we weren't measuring things as quantitatively decades ago as we are now. But now that we are measuring them, we are able to see things like, oh, when it's really hot in a particular year, we might see more of this type of algae or less. Noctiluca, actually, we did not see a lot more of during the marine heat wave here, which was good, but it's not a particularly toxic species, even though it looks really yucky and gets people a little freaked out about red tides. It's not a red tide, so rest assured, if you see a Noctiluca bloom, you can still go swimming and it will be fine. Okay, we're pivoting our way towards nutrients. This is from the Point Williams buoy, which is moored close to shore, but not too far, sort of in between the deepest part of the channel and the shore, near the Fauntleroy Ferry Terminal. In response to the West Point flooding event in 2017, we got some additional funding to buy nitrate sensors, which is really cool. So both our CTD profile nitrate data was started roughly in 2017, but also nitrate data collected at two of the moorings, one of which is Point Williams. So this is looking at nitrate changes over the course of the year, with 2023 highlighted in teal, 2022 in black, and the other years on record in the gray in the background. You can see the instrument precision in the background. That's pretty cool. But one of the things that I like most about this figure is, in addition to the general annual pattern where you've got high nutrient concentrations in the winter and it takes a nosedive during the growing season when algae are slurping it up, and then it kind of goes crazy in the summer as things are being recycled at the surface. One of the things that I really like is the interannual variability. I think this is highlighted best in the winter, where you can see there's quite a range of nitrate concentrations that are observed. This is to be expected because the bulk of the nitrate that we see in Puget Sound is coming from variable sources, so we would expect it to vary over time. Another thing that this highlights is how messy this data is, particularly from the moorings. You see all sorts of unusual data points, like I don't believe that these are real. I don't believe that a lot of these spikes are real. It takes many more people than we have on staff currently to really get this data in immaculate shape quickly. It takes time. So if you are interested in using any of this mooring data, please talk to us first, because if you simply download it from the website, you would get all of it, warts and all. Okay, and that is how I'm going to transition into nutrients, which is the bread and butter of what I do day to day at King County. Think about nutrients. I'm going to start by framing within the context of the West Point failure, which happened on February 9th, 2017. What we're looking at here in this map is fecal coliform. So that is red, big red circle, bad. A lot of fecal coliform, which is not a surprise, I guess, when your treatment plant floods and you can't treat the wastewater anymore. You would expect more fecal bacteria in the water, which is exactly what was seen. A couple of days later, the fecal coliforms have dropped substantially. So the issues at West Point continued for about 3 months after this initial flooding event, before full treatment was restored, but it was reassuring to see the drop in fecal coliforms, even after this short period of time. I was reassured by this. But what other impacts did we see from this West Point flooding event? One of the benefits of having a long-term monitoring program that is already monitoring things, especially close to the West Point outfall, So this is looking at total nitrogen at 55 meters, which is close to the trapping depth of the plume from the outfall. This is looking at 2017 total nitrogen in the orange triangles, the timing of the flooding event roughly marked by the orange arrow at the bottom, compared to the historical data in the gray circles. And one of the things that came out in a report after analyzing all of the data before and after the flooding event was that there really wasn't that much of a change in water quality in terms of what we were able to measure as a result of this flooding event. Just great. I think that's great. But you might be skeptical and say, well, maybe you weren't measuring the right things, which is entirely possible. We can only measure so many things. But total nitrogen is usually a pretty good indicator of what is coming out of the wastewater treatment plants and whether that has really changed, because that is what we care about. But why? Why nitrogen? This is my favorite slide. You'll see why at the end. Okay, so we've got more nitrogen. Hypothetically, in a place like Puget Sound, where nitrogen is the primary limiting nutrient exposed to freshwater systems, where it's often phosphate. In Puget Sound, it's usually all nitrogen all the time. So more nitrogen is more growth, which means more phytoplankton, which means more decomposition, and decreased oxygen. The oxygen is really what we care about, but the nitrogen is how we get to the oxygen. When we have low oxygen, we have sad fish. Okay. So I guess that begs the question of whether nitrogen is changing. We've got this great data set. We can see whether nitrogen changes in response to something like the flooding event, but is nitrogen changing over time? This is data from the Department of Ecology. They also have a monitoring program that samples more places over Washington. So they've got stations out on the coast, they've got stations in Hood Canal, they've got stations in Admiralty Inlet, all over the place, whereas we're just measuring the county. So this is looking at their anomalies of nitrate concentration. I think this is measured against their entire data record, which is roughly the same length as ours. I think it also sort of mid 90s to present. And you can see that it's quite variable over time. You've got some ups, you've got some downs. It might be on an upswing, sort of at the tail end of this, which I think Christopher produced this in 2021. So a little bit of an upswing in 2021. And that's pretty much what we see in our data too. Here again, Point Jefferson chosen for its as our guinea pig of sorts. And this is data from all depths, but each individual depth shows a very similar pattern, which is you've got changes over time, not necessarily a definitive increasing or decreasing trend, which is one of the things that we looked at in our most recent data report. Things are just kind of nebulous. It goes up and it goes down. Is it dipping down again right now? This data goes through 2023. Is it dipping down? It's kind of hard to tell. So what is causing all of these changes or lack of changes in nitrogen? Where does the nitrogen come from? In Puget Sound, the bulk of the nitrate comes from the ocean. This 88% is an older estimate. I think current estimates can go as high as 94%. But it really depends on what is coming. It really depends on what is coming in from the ocean, which is variable over time. Is nitrogen going up in the Pacific Ocean at depth? Maybe. It's dissolved oxygen going down? Definitely. Our ocean source is changing over time. About 7% comes from wastewater treatment plants, and then the rest comes from other watershed sources. But the wastewater treatment plants are the most easily tuned knob on the nitrogen. We can't really control what's coming in from the ocean, but what we can control is what's coming from the wastewater treatment plants, which inconveniently also scales directly with population growth in Puget Sound. So that is just going up. One of the things that I learned when I started this job, again, not having much experience in wastewater before coming to King County, is that 80% of the nitrogen in wastewater is from urine. I thought it was for sure going to be some fecal material, but it was not. It was urine. So it really does directly scale with the number of people, because unless yeah, or pee on your azaleas or other acid tolerant plants. That's what I tell people when they bawl. at the nitrogen inputs from wastewater treatment plants. But yeah, I found that really interesting. But it reinforced, it really does reinforce the idea that unless you remove people from the equation, that wastewater treatment plant input is only going to go up. Oh, man, and we'll get to that. Okay. How do we quantify these wastewater impacts? Well, the first answer is with models. So this is the Department of Ecology's state-of-the-art stainless steel model. What you're looking at here is the domain of the model, more or less. Each of those strangely shaped polygons is one of the grid cells within the model, and the colors are indicating days of dissolved oxygen non-compliance with the water quality standards here in Washington. Oh, where is Seattle? Seattle is here. Here's Seattle. So no days of non-compliance in Seattle, thankfully, but Quartermaster Harbor, unfortunately, out of compliance. And poor Whidbey Basin, highly non-compliant based on this
Just to
model.
clarify, the white is still not compliant?
Correct.
Oh,
Yeah. So any point at which you're seeing not blue is calculated as having days of dissolved oxygen non-compliance with the Washington State water quality standards.
What depth are we simulating this?
oh,
Yeah.
the question was at what depth are the analysis performed?
Yeah, I'm going to answer that on the next slide. But I'll give you a little extra context for how this model is set up. Each grid cell has 10 layers of variable depths. Obviously, the depths out here on the coast are going to be deeper than those in Puget Sound. And the way that the model is run, there is a physical model that simulates water transport and all those things. There's also a biogeochemical model that is like phytoplankton, nitrogen, dissolved oxygen, the works. So it's a coupled physical biogeochemical model. And the way that we calculate non-compliance is that the model is run with pre-human conditions. So that's all human inputs of nitrogen, what have you removed. So that's wastewater treatment plants, watershed sources, everything. And then you subtract current conditions. So the model run with everything. And then you get the oxygen depletion due to human sources. Now, why do we care about that? That's because the way that the water quality standard is written in Washington, earlier I was talking about this number, 7 milligrams per liter. That's the first part of this standard, is this numeric criteria. When you go out into the water and you observe that it's less than that numeric criteria, which can span 4 to 7 milligrams per liter, depending where you are in Puget Sound, then you say, okay, it's less than that number, but is it less than that number due to natural conditions? The dissolved oxygen might just be naturally low, or is that low dissolved oxygen due to human sources? And what they're looking for in that criteria is less than a 0.2 milligram per liter difference due to those human sources. And this can be in any depth. So it doesn't matter if the non-compliance is through the whole water column or just at the bottom, it's still considered non-compliant.
So if the water is below 7 milligram per liter at 200 meter depth, it's non-compliant.
If there's also that additional 0.2 milligram per liter difference. You have to have both pieces for it to be non-compliant.
Oh, I see. The question was, is the compliance related only to being above or below the threshold of 7 milligrams per liter? But no, the compliance is related to both threshold and difference due to human sources.
They call this the numeric and the narrative criteria. The narrative criteria is currently being revised, this 0.2 milligrams per liter, because the way it was written in the code is called the natural conditions provision. I think it's going to be rewritten in a very similar way. So you could probably be assured that it will look like this in the future as well. You might wonder how they planned to assess this. This code is from 1968, before they had really good water quality models. So I don't have that answer. But the way that this is assessed now is through the CLSC model. benefits on the ecology.
And so this assessment is performed by the Department of Ecology, given the data that they collect continuously, the monitoring plan.
Yeah, we also, everyone else who collects water quality data also has an opportunity to submit data, I think every other year, as part of, we submit it to the EIM database that they have as part of the 303 delisting process, because that's really where this goes. Is once waters are determined to be non-compliant, we get put on the 303 list. So a lot of listed waters here in Washington for dissolved oxygen specifically. And there's a lot. You might say, wow, that's a lot, Taylor. You say, well, there's more. There's a lot of questions that spawn from determining compliance and improving compliance with water quality standards. One question is, how can we effectively spend money to mitigate issues that arise from non-compliance with the standards? Another question is, are nutrients and other conditions that arise from increased nutrients actually contributing to changes in hydrotrophic levels? Are we seeing biological impacts of this non-compliance, or is it just non-compliance for non-compliance's sake? And then can we support the water quality standards with better data and better models? Which leads to, what exactly are we doing? What is happening sort of beyond complying just with the permits? What are we doing about nutrients? One thing that we're doing about nutrients is nutrient trading. So this is a current effort that's underway with the Department of Ecology, but also folks at King County and elsewhere. It's still a little hypothetical how it would fit into the existing state code and whether it is actually legal. But it would allow big dischargers to spend money maybe more efficiently than simply upgrading wastewater treatment plants, because those efforts are currently underway. As part of the compliance with this new nutrient general permit, all wastewater dischargers in Washington are going to be required to reduce the nitrogen that is in their effluent. We're aiming for really low standards that would require full tertiary treatment. So employing things like anammox and denitrification to really reduce that dissolved inorganic nitrogen in the effluent. So would it be maybe more efficient or effective, faster to do something like nutrient trading that would allow some of the big dischargers like King County, like Tacoma, for whom implementing this tertiary treatment would be extremely expensive, like billions of dollars expensive? Can we reduce nitrogen input at maybe some smaller treatment plants in some of the watershed sources to get that same effective amount of nitrogen reduction? Can we do that in a different way? And right now, it's not really clear whether this is going to be an option, but people are exploring it as an alternative. Another thing that we're working on is doing some higher trophic level research. So this is something that the UW Puget Sound Institute is working on in conjunction with King County and some other groups at UW, looking at dissolved oxygen thresholds and thinking about exactly how are organisms in Puget Sound experiencing changes in dissolved oxygen. At what point do they have bad effects? Impacts on survivability, impacts on breeding, all that stuff. And then thinking about that in conjunction with dissolved oxygen and temperature to determine the metabolic index. And the metabolic index is something that indicates how much dissolved oxygen an organism needs, given its size, given its species, at a particular temperature. And there are a lot of cool biological models that get employed to make these determinations. And you can then use that metabolic index to estimate habitat compression. So this figure on the right is looking at the change in habitat thickness from a modeling group in Southern California. And you can see that there's quite a change in habitat. And you might see the same things here in Puget Sound, but at this point, you just don't know. That work is still ongoing, but really, really exciting to think about both benthic organisms that are they're experiencing probably the lowest dissolved oxygen there at the bottom of the water, but also more modal species like salmon You know, everyone cares about salmon. That's where all the hearts and the money are here in Puget Sound. We really want to make sure that the salmon are happy and healthy. And the way that we'll do that is in these sort of biological models. And then finally, speaking of models, more models. We are currently funding a couple of different models, sort of doing that do the same thing as the Sailor Sea model, but different. Both of these models were developed for slightly different purposes. So the Sailor Seacast is run through the University of British Columbia, and one of its primary functions in the past has been to predict when phytoplankton blooms are going to occur in the Strait of Georgia. So the Strait of Georgia being in Canada, not Puget Sound, but their whole model domain includes Puget Sound. And so we're working with Susan Allen's group at the University of British Columbia to improve the nitrogen inputs to this model, to improve the dissolved oxygen outputs of this model, to make it more comparable to the results that can be determined by the Sailor Sea model. And then the other model on the right-hand side is the Live Ocean model, which is run through Parker McCrady's lab at the University of Washington. And this model has been primarily also a forecasting model. So looking ahead a day, a couple of days, what are the water conditions going to be like? If you're curious about this, I really recommend going on their website. It's very, very cool. You can see forecasts, you can click on the map and see where particles at the surface are predicted to go. It's really quite special. So we're also working with Parker's group to develop the nitrogen inputs and the hindcasting features of both of these models so that we can look back at a year, 2 years, however many years we want, of model results to directly compare with the Sailor Sea model.
Could you share a link of both of these models on the follow-up email after this?
Totally. And part of the reason that we're interested in having more models is, every model is wrong, but some models are useful, but each model is also probably wrong in different ways. And so by having more models, we can be more confident in the regulatory outcomes. We can be more confident in the effectiveness of our policy choices. And what do all models really need? They need data. And that's where I come in. This is what I do day to day, is I produce data for modelers. That is the primary function now of a lot of the data that we're collecting, is that it is being fed into these models to improve the validation, to improve the results, and just increase our confidence that the model is modeling correctly.
So among those two models that are on the slide, which one has the predicted result closer to the measured observed?
That's a good question. I would say probably the Live Ocean model at this moment,
but still to be compared.
Yeah, still to be confirmed. There's still work to be done. You can get into that if you want. Okay, here are my final thoughts. I have no idea how to. Perfect timing. We're right at 1. And I'm going to run the risk of sounding like a hippie here, but Puget Sound is a beautiful ecosystem and it's complex. It's really important that you preserve it. A lot of people care very deeply about what's happening in Puget Sound. Beyond salmon and orca, everyone wants to recreate in the waters. They want to be assured that Puget Sound is thriving and not on the verge of ecological collapse. So that is part of the reason that I do the work that I do, is to ensure that Puget Sound is not on the verge of ecological collapse. And then finally, sometimes the data takes us in strange directions, and we are surprised sometimes at the ways that it is eventually used. No one would have guessed necessarily at the onset of all of these monitoring programs that we would be feeding all of our data into models, but here we are. So we need as much data as we can get our hands on so we can make well-informed, impactful policy decisions. So with that, that's it. That's all I have to share today. Thanks for listening. You can reach me at this email.
Thank you so much, Taylor.
Thank you.
I will exit and open the floor to questions. By stopping the recording.
Those are quite a bit of questions. Oh, that was our.
They're mapping everything we're saying.
I had a question if no one else wants to jump in.
Great, yes, thank you. Go ahead.
Freshwater limnologists focus a lot on phosphorus concentrations when they think about eutrophication of freshwater lakes. And you talked to us a lot about nitrogen concentrations in the Puget Sound. So I was wondering if you could talk a little bit about nitrogen versus phosphorus and maybe, do you know if there are free living end fixers in the Puget Sound and what kind of contribution they make to the ecosystem?
Yeah, that's a great question. As far as I know, the diazotrope community in Puget Sound is pretty limited, so there's not a lot of nitrogen fixation happening in Puget Sound. We do see occasional phosphorus co-limitation with nitrogen, but it's always co-limitation. We never see phosphorus depleted on its own. Another nutrient that I didn't talk at all about today, but is important to consider, considering that Puget Sound phytoplankton communities are mostly made up of diatoms, is silica. And part of the reason that there's not a lot of phosphorus limitation in all marine systems is that there is simply more phosphorus to go around compared to freshwater systems. You just get more of it. Most of it is geologically derived, but nitrogen is the key here.
Thanks, that was really interesting.
Any questions for the room? I think we were very vocal during the presentation.
I asked the questions for you. Interrupted you.
I have a question. Term data sets that show the benefits, the work that Metro, that's the predecessor to King County. I mean, when I was at UW in the 80s, they showed us like the Setchi dish improvements in Lake Washington. And it's like, see what we've done. Sewers are beneficial. And if there's more data like that showing the good that we have done, since the Clean Water Act came out in 1972, I love stuff like that. When you're extolling the virtues of sewers to an unsewered area, it's really powerful to have data sets like that. And that's what I was hoping to hear when I joined this meeting today.
Yeah, I'm going to tell you good news and bad news. So good news is that you're totally right. In the lakes in particular, that we saw a marked improvement when the sewage was no longer being discharged directly to Lake Washington. However, in the marine system, when secondary treatment was implemented at the wastewater treatment plants, we saw very little change at all. And part of the reason for that, well, the bulk of the reason is that it's something about dilution being the solution to pollution, is that we just don't see much of a signal. Puget Sound is a lot of water, and the wastewater is really just a drop in the bucket. So there isn't a lot of super compelling data that shows a marked change when big wastewater changes have been made, but that seems okay. And part of the concern that a number of people, myself included, share about spending billions of dollars to upgrade to tertiary treatment right away at all of the wastewater treatment plants is that we would not see necessarily much of an improvement. We would model perhaps some improvement in dissolved oxygen compliance, but would we see a lot of day-to-day changes? Perhaps not. One of the things that I would like to see more work done on is for wastewater treatment plants and others that are discharging closer to shore or in shallower depths, what sort of impacts making changes there would have instead. It's sort of an open question.
Okay, thank you.
Thank you. Thank you all for staying a little bit longer with us. I'm gonna off the recording off. And I'm gonna say, thank you for having attended this meeting, and we'll see you next time.
Yeah, thank you. It was a wonderful presentation.
Thank you, Taylor.
Thank you.
Thanks, everybody. Have a great President's Day weekend.